Good morning to all of you. Hope you all can hear me. To start my section, and I will show you the content I'm going to cover in a bit. But before that, I think it's better to get to know all of you. So let me quickly share like a small poll. So please respond. And my class, I would like you to be as interactive as much as possible. So therefore, you can use chat box, or you can raise your hand and ask questions if you have any doubts or if you have any comments. And I will upload some content to your PGLMS, so you can access content from there as well. Okay, so let me launch the first poll. I'm good that 80% of you responded. That's like a relatively higher number, 85, even better than in a usual class. Okay, so let me share the results, and based on your input, so it seems like 40%, the majority studied the area beyond whatever the fields I mentioned earlier. So we have a significant amount of biomedical people and biology, in addition to other degrees. So if you're coming from like a completely different background, and if you have any issues, so you can always contact me, or ask class questions during the class. So that way, all of us can move in the same phase when we move forward. Okay, so that was the poll. And then to introduce myself, as you already know, I'm Venura Herat, and so I'm a molecular biologist by molecular geneticist by training. And also, when I don't do molecular biology stuff, our favorite thing is to go and enjoy the nature. And so I'm on I try to involve in scientific communication. So because that's one of the issues our society is facing, especially now. Things happen in the US and other places. So there's a lot of misinformation, disinformation, and every time you open your Facebook, it says oh, there's a miracle cure if you eat Nihagala or something like that, and on a nightshade, and those kind of things people fall. And then as scientists, not only me, you as well. So the most important thing is to disseminate science and make decisions based on evidence, not basically because someone said so or something like that. So therefore, I try my best to involve in scientific communication. And other than that, so we travel, and I love bird photography. And you can see this, I was taking a picture of a common kingfisher in the Hubbard Lake, and my wife captured it. So anyway, so so remember, studying is not the only duty as scientists. So your responsibility is to pass the correct information. Now you're taking molecular biology or the and bioinformatics related stuff. It's very important, because every time when people hear, okay, so GMO, people will freak out, but they don't realize that. So we eat a lot of DNA protein every day, like 3 meals or even more than 3 meals, and still we can digest them. And there's a lot of misinformation, because people think genes are some foreign thing, although you have it inside your body. Anyway, so moving forward, so my research is focusing on mainly on stress, how different environmental conditions affect the metabolism and overall survival of plants. And in addition to that, I involved in other projects beyond plants, from mushroom to leopards to bees. These days we are gene sequencing all the honeybees in Sri Lanka. There are three. I don't know whether you know. There are 3 honeybees in Sri Lanka, and we sequence all of their genomes. And then so soon we will publish that paper. And it's really interesting. So that is the biggest advantage of being a molecular biologist, because you work with DNA and RNA and protein. So those things you found in any living organism plus viruses. So therefore, you can do. I mean, there are no restrictions. It's There's no what do you call this, border, so you can cross any fields, because I know many people, like even my colleagues, so they started as plant biologists, so now they are like working at UCLA doing biomedical research. Okay, so vice versa. So you can do that. Okay, so there's a question. Okay, so like a good question. So from Sashini, so regarding the software I use, yes, I use BioRender. Unfortunately, Peradin is not giving the license to BioRender, but since I'm collaborating with Texas A&M University, through them, I have the access. And yes, I really like BioRender. And also, if you want something equivalent, but that not that much resources, it like you can use Canva. Canva is also a very powerful platform. I use BioRender and Canva both for making all the graphics I use. Okay. Anyway, moving forward. Uh, so as I mentioned to you earlier, so I will be covering 8 hours of lectures, and that includes genomic data processing, data analysis, gene expression omnibus, data deposition, search, retrieving, and integrate of data, as you can see on your screen, data flow, and then the most fun part is how you can process biological data and how to assemble genomes and annotate them and then linking all those data. So today we are going to focus on gene expression omnibus, but next week we are going to talk about genome assembly, and during your practical, so there we will do all these practical aspects of things we discuss, and I will give you your own genome to play with, and then end of the class, the practical, you will be assembling your own genome. I mean, not your own genome, but you will get one specific genome for catered for you. So that way, you can get a very nice experience at the same time. So when you submit your the activity, so that none of your results will be the same, because I'm giving you a very unique genome customized for each one of you. Okay. So moving forward, so let's assume a situation. Okay. You're as a molecular biologist, you're kind of playing with bacteria, and then accidentally, so there's let's assume there's oil, some let's say mineral oil, some oil vial with you, and then you spill it on your bacteria by accidentally. Okay, so what you realize is, surprisingly, some bacteria died, but some bacteria survived. So now this is really cool, right? Let's say it's tau or something like that. It's very not good for environment, environment unfriendly thing. And now you have something. So therefore, you're thinking as a molecular biologist, I need to get this and move forward. So you're going to work as a scientist, and then you're going to basically screen that bacteria, so to see what unique feature inside that bacteria. What you're going to do, you're going to do something like this. Okay, so let's see whether
by agitating the tissue, steel balls, or beads, and a licensed agent.
Yeah, so you use a genome grinder, and then you break the cells, and then you make a design an experiment with the oil, without oil, and then you isolate the gene RNA, and then you look at the expression. Okay, so it's a pretty cool thing going on, and you generate a lot of the reads with all this expression information, and then you say oh, I found an immortal gene, because we can't. I mean, you can't kill this one without with all this dirty oil. So therefore, you will become very famous, and then you're going to win the Nobel Prize. Okay, so now you're very famous, and your research findings outside. But the thing is, so let's say a scientist from Peradeniya University, or let's say Harvard University. So now they're trying to recreate your results, because they also want to check whether they can use your specific bacterial gene to create the same scenario where they can use it for, let's say, bioremediation to treat all these the contaminated lands. So now when they try, I mean, they're trying and trying and trying and trying and trying, but it's not working. And it can lead to something like this. I don't know whether you remember from last year. So you heard about the room temperature superconductors, and like one Sri Lankan scientist, so Sri Lankan born scientist, and he became very famous. And unfortunately, so none of his the findings were not reproducible. Okay, so that ultimately led to they basically get rid of him from the Rochester. Okay, so this is the situation. And why I'm talking about it, probably you're wondering what this has to do with anything. Okay, so because when it comes to science, one of the major things we consider is the reproducibility. Okay, so when you do a research, it should be reproducible, because anyone can do one run or even fake, and then say, okay, so this is a miracle cure, or this is the wonder. But in reality, if you can't reproduce, that is not a good science. So therefore, the scientist will not basically going to recommend whatever you found unless it's reproducible. So this is something you must remember, because you will be doing some projects. And when you do your projects, make sure that your data is reproducible. Otherwise, I mean, the same thing had happened to you, like what happened to Rangadayas. So you will become very unpopular scientist in the whole world. Okay. So therefore, it's very important to make your data, or your findings, reproducible. Okay. So let me quickly share another poll. Okay. So this is a small poll to see whether you're familiar with the data source I'm going to talk about. Okay, so please take a minute or so and answer all the questions, and then we can move into our very interesting topic. So let me end the poll, and we'll share the results with you as well. It's a very nice normal distribution. So we have most of the people are somewhat familiar with the NCBI. And most of you didn't realize that there's a very nice tutorials and other things available in the NCBI and the training site. So that is a very good resource for you to learn, because even though sometimes even though you're expert, you don't know in every aspect of NCBI. So therefore you can even learn even more things if you use those resources. Okay, so those who are not very familiar with NCBI, don't worry too much about it. So we will it is easy. It is made in a way that anyone can get familiar with it and then use its data. Okay, so let me stop sharing, and we will move into the main topic of today. So that is let me go back. The Gene Expression Omnibus. Okay, so it is basically your getaway to genomic data, because it contains so much resources. I'm going to show you in a bit. And if you think about think about the data available in the geo, it's definitely more than the stars you see on the sky at night when you look at it using your own eyes, naked eyes. Okay, so therefore that's the amount of data available, because I am pretty sure maybe some other teachers, they talk about the Moore's law, right? Because initially our main issue was generating genomic data. It was so expensive, but with the advancement of technology now, you can generate data gigabytes or terabytes of data within a couple of hours. So now we have a problem of storage, because we don't have enough storage, and also we don't have enough manpower to analyze all the data. So therefore, let's say you decide, okay, you're going to stop doing any other work and start working on whatever the data available. I guarantee you, your lifetime will not be enough to analyze all the data available in the Gene Expression Omnibus, or any other NCBI database. Because I remember, when I was doing my PhD, so we generated a massive data set, and then my boss told me, okay, Venura, you know what? I can retire today. So that's what he told me in, I think, 2011. So it's I can retire and only work on the data set we generated for the rest of my life. So that's what he said. And so that's the beauty of this massive amount of data we generate nowadays. Okay, so the Gene Expression Omnibus contain all that data. So now you're wondering, those who are especially not very familiar with the Gene Expression Omnibus, what is this GEO? Okay, so this GEO is a public repository, okay, and that is serving as an international archive. Okay, it's not restricted to Sri Lanka, or I mean, it's US based, so it's not only to US, but people from this small country, Sri Lanka, we can also access that, where it contains a lot of high throughput genomic data. Okay, and if you think about it, the analogy will be, it's like a library in Alexandria. I think you heard about the library in Alexandria, right? So if not, please read. But unfortunately, it's a very there are so many myths around the Library of Alexandria. So why it was destroyed, and how it was destroyed. But this one, it will not burn down, and it is constantly growing. It's a massive archive, but now there's a threat with the new US administration. They are cutting funding for NIH, because NIH is hosting NCBI. So therefore, there is a threat very similar to the Library of Alexandria. We sincerely hope such thing will not happen to this database. Okay, it contains 238,316 public series as of yesterday, and then imagine 7.45 million samples, and more than 84,000 people submitted their data to the NCBI, including I'm also a part of it, and I'm proud of it. And also it contains information for of 6,681 organisms. So now you can see the amount of data it's carrying. Okay, so to dig more into the GEO, as I mentioned earlier, it's a public repository, and what it's doing is it's archive and freely distribute the data related to microarray and also the next generation sequencing platforms. I think that is something you learn from the other the instructors. The next generation sequencing, they are second generation, this is a third generation. So likewise, so all those data will be stored in GEO. But remember, GEO is attached to SRA. Maybe you will. We will talk about SRA during the next lecture. Sequence Read Archive, SRA, and also, so it contains some other forms of high throughput functional genomic data. Okay, so therefore, in very simple layman's term, it's a public repository contains a large amount of genomic data. And if you think about the evolution of Gene Expression Omnibus, it was launched in year 2000 by the NCBI. And then once it was established, most of the journals are basically put a condition that if you want to publish, you must deposit your next generation sequence or the high throughput sequencing data in this archive. Okay, and then so they realized, okay, there are a lot of data coming in. They wanted to have some kind of cataloging system and structured data system. So they introduced data sets and profiles. That is something we are going to discuss in a bit. And in 2013, they came up with a platform. Why not data? So let's us provide a way to analyze the data inside that system as well. That's gave the birth to GEO2R, where you can basically grab data from GEO and analyze using our platform. And by 2014, there are 238K series, and as I mentioned earlier, 7.45 million samples, and it is growing exponentially. So that is not going to change in recent times. So you will see a peak I'm going to show you here. See, so how it's growing. I mean, it's somewhat reaching a plateau, but you never know. But still, it's growing almost exponentially. Okay, so let's see whether you guys were not sleeping. I have quick question. Where is my pulse? Okay, so I'm going to stop. No, more answers are coming in. Okay, so I'm ending the poll. Okay, so let me share the results. 70%. I'm 100% sure that you're awake and listening. And then there's for 9. The remaining 30%, you you're not paying attention, or maybe fell asleep, or you're thinking about maybe using your phone. Anyway, so it's the year 2000. Okay, so you're moving forward. Let me close it. Okay, so I think now you have an idea why this database was established. If you look at the original goals of the Gene Expression Omnibus, is to provide very, very comprehensive and versatile database. So therefore, we can efficiently store high throughput functional genomics data. And if you want your database to be successful, so one of the major thing is it needs to be what you call a very, very user friendly. Even anyone can use it. So therefore, they had the goal of offering a very simple submission procedure and the formats that support complete and well annotated data deposits for research community. Okay. And also not only submitting, but also it should provide a very user-friendly ways to search, identify, review, and download these gene expression profiles. Okay, so those were the 3 major goals so they had when it comes to establishing this database. And they're pretty successful with their goals. Okay. Okay, moving forward. If I'm going too fast, let me know. If I'm going too slow, also let me know. Okay, so when it comes to okay, there's a question. Give me 1 second. Okay, so very good. So there's a question. What is the difference of this database from other data sets in NCBI? So I'm going to show you very clear one difference, and then I will explain other differences. Okay, so when it comes to NCBI, it's one big entity. It contains various databases. Okay, to give you an example, so there's another database called SRA. SRA means Sequence Reads Archive, where all this the data we generated, the raw data, we store it in SRA. So therefore, GEO contains expression data, how much genes are expressed. Okay, SRA contains all the reads, the sequencing reads. So that basically showed this expression change. Okay, so those two, these two databases, they are highly linked. If you think about the nucleotide database, it contains, let's say you do a barcoding, you identify a species, what you do. So you usually use mitochondrial markers like METK or something like that. And then you basically deposit your sequences in the nucleotide database. And then genes, there's another database. So here, Gene Expression Omnibus stands for the gene expression data. So therefore, it is different from other databases, but it is interlinked. It's highly closely interlinked with the sequence read archives. Because to give you an example, when you submit your data to the Gene Expression Omnibus, you can't deposit your raw data into the Gene Expression Omnibus. Okay, the read information. So what happens is, when you can go to GU and do the same, what they're doing is, so they take the large amount of reads, because they are one file is like 5 gigabytes. So they will automatically transfer them into sequence read archive, right? And they keep the expression, the outcome, in the Gene Expression Omnibus. Okay, so these databases serves for that specific purpose. So therefore, when you think about the expression data, so you are going to look at not only Gene Expression Omnibus, but also the sequence reads archive, depending on your requirement. Because these days, one of your senior, maybe one, senior batch student is working with me, and we are looking at the microRNA expression profiles when people exposed to COVID. Okay, so what we are doing is so we can get the expression data directly from the Gene Expression Omnibus, but instead we are doing a meta-analysis. So therefore, we are getting all the raw reads from the sequence read archive, and then we are analyzing them in order to see across studies how these microRNAs are expressed. So I mean, that's depending on your requirement, like I mentioned earlier. So you need to decide what database you are going to use. And there's very nice help available for each database, as I mentioned earlier, the training material. So you can when you get a free time, so maybe don't look at TikTok, but look at the NCBI. In they have videos on YouTube. They uploaded by NCBI, and you can look at them for a change. Okay, so when it comes to microarray data, remember I told you it contains next generation sequencing data as well as the microarray data. I mean, I had the chance to submit both types, because when I was doing my PhD, microarray was the big thing. The next generation sequencing was starting. And then I had the chance to work with the Illumina sequencing. And then we uploaded that data into the sequence read archive as well. Okay, so the microarray data, they're basically stored in GEO, because microarray data is not that big. So they are 510 MB compared to RNA-seq data, as I mentioned earlier, one file is uncompressed 5 gigabytes. And there are some special circumstances where some RNA sequencing data sets, they are deposited in GEO without raw data available in the Gene Expression Omnibus. Okay. And so now you're thinking, okay, so this is basically the US base. Luckily, we have some alternative ones as well, just in case. Remember the Library of Alexandria? We don't have it to the NCBI. So when it comes to the Europe, so there are a lot of people involved in this. There's what we call as the European Bioinformatics Institute, or EBI. So they have their own equivalent of these databases, ArrayExpress and ENA means European Nucleotide Archive. So we have those. So those sites also contains expression data and then the raw read information. And those are the two major databases, basically duplicating the same function. And there's one small one. Oh, this is the ArrayExpress, as I mentioned earlier. And so this is basically catered for the European researchers or investigators, but also we can, I mean, we don't have one like in a Sri Lankan one. So therefore we can submit either to NCBI or ArrayExpress. And there's another one, what we call as the DDBJ, or the Japanese one. So they have what we call as DRA. Okay, and it's relatively small compared to NCBI and ABL. Okay, so there you can deposit your sequencing reads. Okay, so now you're wondering, okay, so there's NCBI1, EB1, DDBJ1. So this is going to lead to issues and confusion because these three databases serve the same purpose, but they have their own cataloging system. So therefore, when you search, there's a big chance you'll get lost. Okay, so therefore, what they did is, so they decided, okay, so we need to do something about it. So therefore, we need to integrate data sets across different platforms. So it gave rise to what we call as the International Nucleotide Sequence Database Collaboration, or INSRDC. And the objective behind was to ensure that all public domain nucleotide sequence data that are deposited and archived, preserved as a part of scientific record, and is accessible in standardized formats across the three sites during daily data exchange. Okay, so they communicate with each other daily, and then they have reference to the other database. So that way, they even though you search in EBI, still you can link it to the information coming from NCBI or DRA. Okay, so therefore now it's more streamlined compared to having three independent databases. Okay, so that really helped scientists. And then what you see in this, the inside this triangle, so it's what we call as the trace archive. Okay, so the idea is, remember, we are depositing data, more than what they can link. So therefore, there is some delay, so that we can't avoid. But eventually they will catch up. But so whatever you found in trace archives, they are already cataloged and linked across databases. Okay, so you have any questions regarding Gene Expression Omnibus? I'm not finished, so I'm just starting. And as I mentioned earlier, you can ask questions anytime. So you can also use the chat box in order to post your questions. Okay, so we are going to move forward, and then so let's look at these databases for a change. Okay, so let's take a ride. So I will start with let's see. Okay, let me move the screen to this side. All right. So as one of you very correctly asked, there are many databases. See, but our interest on the Gene Expression Omnibus. Okay, so you see there are two different types. One is called the gene data sets, and the other one is called GE profiles. So you can search from here, but instead you can directly go into the Gene Expression Omnibus. You can see they basically categorize their resources depending on the use. So here we're interested in genes and expression. Okay, so then you can see, so there are a bunch of databases available for this specific purpose. Even if you want to go further in, you can click this database tab, and either way. So here you can see Gene Expression Omnibus. Okay, so this is the big one, the overall one. Okay, so here you can basically search anything. Let's say if I want to search COVID. So usually it will give you how many queries. Say there are 19,711 results. So likewise, so this is the landing page of the Gene Expression Omnibus. You can see. So there's ways to query, and remember GO2R I mentioned earlier. So you can analyze, you can search, and you can. There are different ways to access. We are going to discuss those ones as well. Okay, so this is like just overview about the site, and then let's quickly look at the sorry. Okay. Okay, EBI, European Bioinformatics Institute. Okay, so here you can find data and resources. Okay, so they have different ones. Okay, explore. Okay, so you can see that similar to what you saw in the NCBI. So there are various ways, but the different format. Okay, so what we are interested in is okay, so you can see the expression levels, for humans, for even you have plants, fungi, and these are basically curated data, and then you can access the experiments. And also, if you want to see the European Nucleotide Archive, let's see. Searching is lazy. ENA. Okay. So this is the Expression Nucleotide Archive. Sorry, European Nucleotide Archive. Okay. So you can submit your data here. Okay. So that is EBI, and the Japanese one. Remember the DRA. It's very small database compared to the others. Okay. So DDBJ, and so they have overall sites. And I don't use DDBJ, but let's see what are these. Here you have the DRA, and then completely different interfaces. But remember, with the collaboration, they share data. Okay, so those are the three major places we can get that kind of information. Okay, so moving forward, so the question is, now the data is available, but is that enough? Okay, because remember I told you about the earliest the situation where the Ranga dies, so you're finding a Nobel Prize winning bacteria. So the thing is, you can generate data and then you can deposit data remember when we store things, sometimes we forget where we kept it, or and sometimes we put some kind of a reminder, but you have no idea what the reminder is for. So that kind of thing can happen, right? So the availability is not it's important, but there are some other important when it comes to that data sets. Okay, so the scientists realized when people started depositing, so they are depositing without what do you call kind of the data was all over the place, because the naming and the treatment conditions. So someone given, someone not given, some are all over the place. So therefore they decided, okay, so this is not going to work. Making available is a must, but it should be usable as well, because you can just put data and then put 1 2 3 4, and you have no idea, especially when you do lab research. Remember sometimes we label tubes as 1 2 3 4, and then you put it into the minus 20 freezer. After couple of weeks, when you look at your the box, you see okay, 1 2 3 4 1 2 3 4, and you have no damn clue about what those tubes are for. So I mean, same thing can happen. So therefore scientists came up with two systems, okay, to overcome this problem. One is called Miami, the other one is called MinSeq. Okay, so the Miami, it came first because the microarray came before the next generation sequencing. Okay, so the long name for that is the minimum information about a microarray experiment. So that means when you submit your microarray data to the archive, so you have to have some basic information. What kind of samples? What is your experimental design? What kind of hybridization? Because there are different microarray platforms like Affymetrix. You have then like the other one like the Agilent and other ones. So they are use different hybridization chemistry, and also there are different array designs. Okay, and and when you submit, you need to put all those information in addition to the raw. I mean, and it should be included in the raw data, and then you also need to upload the process or the normalized data. Okay, and the MinSeq is for the next generation sequencing. So there you have to mention, okay, what kind of biological system you're using, samples, experimental variables, and you need to add both raw and process data. And then you need to provide general information about the experiment and also how you processed your data. So that way anyone can reproduce your output. Okay. So those are the two major kind of data standardization system scientists came for. So now, let's imagine, when during your project, if you're generating next generation sequencing data or microarray data, at the time you submit your data to database, there are some minimum requirements. Unless you fulfill that minimum requirement, the database is not going to accept your data archive request. They will reject, and then they will send back, okay, so there are some information missing, and please attend to that. Unless you put it into the archive, you can't publish your study. So therefore, you must stick to Miami and MinSeq guidelines. So let's quickly look at well, it's 8:50, so let's take a small break. So probably you're feeling tired. So we will take 5 minutes break. Now it's 8:50. We will meet at 8:55. So you can have a tea or something like that and come back. And then we will continue. Okay? Okay. So I guess you're all refreshed and ready to know about the Miami data requirement. So we will start with the Miami as we discussed before the break. Okay, so when it comes to this, the requirement, it should your data should contain the raw data. Okay, when it comes to microarray data, either you get output file as cell file or GPR file, and you need to provide that raw data files into the archive. And also you need to put the final process, that means the normalized data for the whatever the hybridization you did for your experiment. Okay, so that in very, very layman's term, that is the gene expression matrix you use to obtain a conclusion from your study. And the third thing is, so you need to provide the sample annotations. It is essential, and that should include the experimental factors as well as their values. Example is given here. So let's say you are doing like a drug trial. What kind of a compound you use, and what is the dosage you use. Okay, so you need that both the informations are crucial, so that information need to be provided when you submit your sample. And the third is the experimental design, and also you need to show what are the sample data relationship. Okay, so the what are the controls, what are the technical controls, what are the biological controls. Likewise, so those information should be included. And also it should carry sufficient annotation of the array you use. Okay, so because I don't know, I don't think I have microarray slides with me. Microarray slide is something like the slides you use in microscopes. So the difference is this it's this size, like it's same size as the cover, not the cover slip, the slide. And the slide, the one I use one for eyes. Like each slide contains 43,000 dots. Each dot represent a gene, so basically. To represent one specific, like each dot represent one specific gene, and there were like 43,000 dots there. So, and then like now you're wondering how you identify, like if you look at my background picture, so when you develop a slide, it will look like that. Okay, so there is like 43,000 dots with different colors, like a Vesak Toran or something like that. And the thing is, so they are in blocks. So we know, like, let's say block 2, let's say 300 dot corresponds to this specific gene. So likewise, that information needs to be provided so then others can correctly use it. Because even when it comes with microarray chip, the same chip, with time they upgrade it. Okay, so there's like version 1, version 2, version 3, likewise. So if someone use just thinking about the slide, if they use old version, so their results is not going to be correct. So therefore, it is very important to provide that information. Okay, and also we need to, we need to provide the essential laboratory and data processing protocols. What tool or algorithm you use in order to analyze the data, and then all the specific settings or parameters you use, those need to be provided with your data. So this way we ensure that your data can be reproduced by someone else. Okay. So that's what we call as the Miami for the microarray. And let's quickly look at the MinSeq guidelines. Okay, so here we are basically submitting the high throughput sequencing data, especially like next generation sequencing data. And here we need to include the description about the biological systems, samples, and also the experimental variables being studied in this one. So like the earlier case, what is the compound, what is the dose, and antibody, whether it's a chip-seq, what kind of organism, what kind of tissue you use, and what kind of a treatment you apply. So that information you need to provide. There are templates, so therefore you don't need to worry too much. The only thing is you need to include the correct information in this specific template. Okay, and also it should contain the sequence read data for each array. So, and it includes all the read information and also the quality information already embedded in FASTQ format. And then the what kind of a scale you use for that the basis selection. And also you need to include the final processed or summary data set to assay this specific study. Okay, and then you need to include general information about the experiment, and then the relationship between sample and generated data. Okay. And then the final requirement is you need to include essential experimental data processing protocols, very similar to the MAMA guidelines. How your the nucleic acid was isolated, purified, processed, and how you sequenced it, and what kind of instrument you use, and what kind of labeling technology you use, and what kind of alignment algorithm and data filtering, and all this information needs to be included under this MinSeq guidelines. So then if you satisfy all these 5 requirements, the Gene Expression Omnibus and SRA, in database will accept your data. Okay, so that's how it happens. Okay, so those are the minimum requirement. Don't forget, says minimum under both situations, because you need to provide at least this. But if you're a good scientist, you provide even more information. So because sometimes we get greedy, right? We don't want someone else to because every time you generate one data set, it contains so much information. You can publish many papers. So therefore you tend to keep some information with you, so that I mean, some you can, but you need to provide that minimum requirement, because otherwise it is not reproducible. But I'm not encouraging you to keep secrets. So rather, science is open thing. So that's what's happening here in Sri Lanka as well. Sri Lanka, people so especially policymakers and administrators, they are so greedy when it comes to the data, and doing working on this the organisms found in Sri Lanka. They say we are not going to sequence them, and we are not going to make them available for the rest of the world. But the sad thing is, we do bioinformatics here because others made their data available. Okay. And we can't say, okay, so you should make it available, but we are not. So that is not ethical. Okay. So therefore, that kind of a mentality is not going to take us anywhere. So therefore, remember, so you can't expect other person to give, and at the same time, you are not going to give anything for them. So it's not fair exchange. So therefore, I think when you decide things, so you need to consider these kind of things as well. Okay, so let's look at the gene expression omnibus architecture, because now we are talking about the data submission requirements and the outside appearance, but we need to know the structure of gene expression omnibus. Okay, so there are what we call as the four musketeers, and I'm pretty sure some of you watched or read the book The Three Musketeers. And so here we have platform records, we have sample records, we have series records, and we have data set records. So those are the four kind of pillars of the gene expression omnibus. Okay, and so the at the bottom of each picture, there's a very very simplified version of an explanation about the each component of the geo architecture, as you can see. Okay. Platform is like a recipe. Okay. And sample records, it's providing its own information, each sample. And series combine multiple things. It's like a book containing all the chapters of your story. And the dataset record is very handpicked. You will see very handpicked analysis ready datasets. Okay, so those are the major components. We will look at each and every in detail. Okay, so let's see. No, there are no questions, so we are moving forward. Okay, so the overall structure is like that, as I mentioned earlier. So you have platform, you have sample, and series. These ones are basically provided by us. Okay, and data sets and profiles, so they are coming from their side. Okay, or the NCBI the scientists, they do that part, or that's how they curate, and the first three is coming from us. Okay, so when it comes to the platform, it contains a text description about the array or the sequence used for the whatever the experiment scientists like you carried out. And it will basically give you a tab delimited a table of the array template. Okay, so array template means, say, remember I told you this microarray. So the basically the platform contains, okay, so this is Affymetrix array, and then so this is the how the data points are distributed. Data in the sense the dots. Okay. And when it comes to the samples, it contains a text description about the biological samples. and what kind of a protocol was followed and what kind of a treatment it underwent. Okay. And the bottom, like, similar to the text tab limited array template. So here you have the process hybridization results. Okay. And if the raw data is available, so that will be there as well. And then so you have like the bottom, it's like downloadable sequencing files, like it says, right, raw data, process data. Likewise, we will visit a site and I'll show you all those things. And then the third one, the series, and it will give you an overview about the overall description of the experiment. And it contains tar. Tar means so basically compressed archive of original data files, and also the process data files. Okay, so that's how the user side of the NCBI Gene Expression Omnibus interface looks like. And when it comes to their side, so this is basically a hand-picked set of data analyzed by the NCBI staff, and then it contains the corresponding gene expression profiles coming from that specific data set. Okay, so this is basically the full summary of the NCBI interface. Sorry, Gene Expression Omnibus interface and its different components. Okay, let's see, no questions. Okay, so now we will look at individual aspects of it. Okay, we will start with the platform. Okay, so let's see what is a platform. As I mentioned to you earlier, so this is basically the system they use in order to generate the data for that specific experiment. Okay, so this is basically, it can be a chip you buy from the vendor, or it can be the sequencing platform you use in order to do the sequencing part. So therefore, this is basically, as name implies, it's a platform used for the data set, as you can see here. So it contains, like, the link to the manufacturer's website and what specific. Here it's a Affymetrix array. So therefore, like, they are like organism specific, and also, like, what kind of probes are there. And see, remember I told you, so they update like, time to time. So therefore you see like in different builds. So you need to, like, pick the correct build, otherwise your information will be different. Okay, and then the company details, and at the bottom you will have, like, samples and series. Okay, so the platform can be common for, like, many experiment, because the company sells the chip and then you buy it and then you do experiment. And not only you, probably your neighbor is also doing the same, and your friend in, like, States or Europe is doing the same. So therefore all the samples and series use that specific platform is given below at the bottom. Okay, and as I mentioned to you earlier, the platform contains a summary description. It contains information about the array or the sequencer used for that specific purpose. And there will be a data table that you can download and define the array template. Okay, and the most important thing you need to know is, like, all this platform, the series or the data sets, they have their unique accession number. So that means they when you publish things, or, like, when you analyze things, so you're not going to miss it. Okay, so you always know, okay, so this is the specific accession number. So therefore it's very unique for each platform, each sample, each profile. Okay, so that's how they maintain a uniform system across the globe. like across the world without any mix up. Okay, and if you can remember, as I told you earlier, that this platform may reference many samples submitted by many submitters. Okay, so that information, because they use the same platform. So next we will quickly look at this specific example of the site. Yep, right there. Hope you can see it. Okay, so this is an old, this was submitted in 2004. Okay, so you can see there's a description, and then you can see. So this is an alternative for like different platforms, because if you look at the name, it starts with GPL. So GPL specifically stands for platforms. Okay, so therefore, like this platform is an alternative for the many other platforms. And here, so these are the different the headers, like columns included in this the probe, because remember that each dot is a probe, so that probe correspond to different information. You can see all that information here. This is the probe ID, and this is the gene bank accession number for that, and it will show, okay, so this is for the mouse, and so what kind of a sequence, and then like what kind of a target. You see all that information is given here, so therefore you can very easily refer this specific one, even the gene ontology. Okay, I think you learn about the gene ontology, right? And we will talk about gene ontology when we talk about the genome assembly next week. Okay, so likewise, all that information is available. You can download the full table from here. And also you can get it from like different formats, the soft format here. And all the way bottom, you can see, like, by clicking this, you can download all this, the information to your computer. And then you can do the analysis. Okay. So this is how, this is what platform looks like. So let me quickly something. Let's try something else. Let's just search, let's say these days, measles. Measles. I don't know whether the spellings is correct. Measles. Okay. Okay, so what I wanted to show you is basically to show you different platforms. Okay, I just wanted to show you, like, the Illumina platform, because earlier we are looking at the Affymetrix. Affymetrix is so basically for the microarray style. Okay, so you can see here. So this is the Illumina Nova 6000. It's a little old because now we have much newer versions of it. Okay, so so now you can see this the header and everything it changed, and so you don't see, like, description like this probe ID and other things, because this is high throughput sequencing. Okay, so therefore, like, depending on your platform, it can be PacBio, or it can be Nanopore, like the generation one. It can be Lumina, and then BG has their own system. And then what else we have? We have a Thermo one, what is called the one you have it in Credence. I forgot the name. Iron Torrent. So those are like different platforms. Okay, so you can just search the Gene Expression Omnibus and try to see other platforms. Okay, so the next part is the samples. And as I mentioned to you earlier, it contains the description about sample and then who submitted it. Okay, if you submitted it, so your name will be there. Okay, and then it will show you what platform it belongs to, and then what, whether it's a part of a specific series. The series is given here. Okay, so and then like someone, if they reanalyze it, so then so you have that information there. And here, so this is array based one, so therefore you have the amount, like the expression level, and then what is the confidence or the statistical significance, whether the p-value is like high or low depending on your the gene's expression. Okay, so when it comes to samples, the record, it includes the conditions under the specific individual sample was handled, and if there are any modifications done, it also needs to be mentioned. And then the abundance measurements for each element derived from it. That's the like how much, or like what are the levels it was present. The abundance measurement is given there. And also, very similar to the platform, it has its own unique and stable accession number. So remember, this is very important: unique and stable. Okay, so this one is unique. So therefore, only, I mean, like this, whatever the GSM number is specific for this specific sample, it's not going to change. And also, stable means it will be there like that forever. Okay, so they are not going to basically modify it under any circumstances. Okay, it will be available hopefully until the end of time. Okay, so the most important thing is your sample entity must reference only one platform. Okay, you can't have one sample representing two platforms. It's both practically and theoretically impossible to have that kind of thing. Okay, but your samples can be included in multiple series of data. So that is possible. Okay, so let's quickly look at an example. Okay, so this is an example sample. So this is submitted in like 2004. All of the samples I use are related to the measles, because these days measles is a preventable disease. But unfortunately, because of all this misinformation, the measles is all over the US as well as in Canada, because Canada eliminated measles, but somehow all these anti-vax stupid people started spreading all these lies about COVID vaccine and other vaccines. Now we have a public health danger because of this misinformation. And so this is going to cost us dearly, because we are like very healthy because of our childhood vaccinations, and then like the subsequent one. But like when we stop doing that, what is going to happen? So we are making the whole society vulnerable for these preventable diseases. This is very wrong, but unfortunately, all these the Facebook heroes and TikTok heroes, even Sri Lanka, there are so many anti-vaxxers spreading misinformation, like P.V. Vijayawardena, and then like this some one is like agriculture guy, and the other guy is from like he's an engineer, so he has no damn clue about how like vaccines work, because he thinks, okay, I survived because he survived because of getting COVID, so others should also survive. That's what we call as survivorship bias. You can read about it. It's a very very interesting like phenomenon, because you think others will also survive because you survive. So this is like the World War II story is behind this, because like they have all those fighter jets, and fighter jet some came back after like going into the fight and getting all the hits. But the thing is, so none of them got critical hits on their like engine, or like the fuselage. So therefore they survive, but doesn't mean like, the next plane going there is going to survive. So that's what we call a survivorship bias. So therefore, like, these people, they are spreading all this misinformation, and they are risking all of us. Because if you can remember in 2021 or like in late 2020, some people, this ivermectin and the other people decide, okay, so there will be a herd immunity for COVID. So now it's 2025. Do you think we have a herd immunity? Not necessarily. So therefore, like, don't listen to all this crap, um, because measles, mumps, measles, and rubella, you can easily get that vaccine and then get yourself protected without getting all this the negative impact of the virus. Okay, and same for the COVID. Unfortunately, we don't have access to COVID boosters because of there's no vaccine equality or equity across the world. So therefore, because if you go to Europe or state, you can get a flu vaccine with COVID boosters. So that way you can protect yourself. You should see the long COVID symptoms and how much agony there are people going through because of the long COVID. So therefore, make sure to wear a mask and protect yourself. Anyway, coming back to the sample information, as I mentioned earlier, so you have the submitters information and also the values, the expression values of different the genes you find in your sample, and then you can download those information through FTP or HTTP, or just clicking it, or you can use the FTP client like FileZilla or Duck. What Duck? I can't remember. Some Duck and Aspera client. So that way you can download your data. Okay, so that's about samples. And next, we are going to talk about the series. Okay, so this is a series. Okay, so when you look at a series, you can see, so there's a title, there's an experimental type, and then the overall summary about the experiment, and then how the design, and also where it is published. All that information available under a series. So the idea of series is it's linked together a group of related samples, and it provides a focal point to description about the whole study. So this is actually the book that is showing what are these samples there for, and what kind of a experimental design we use, and what is the outcome, and where it's published. That information is there. And it can also contain a table describing the extracted data and also the summary conclusions made out of these data sets, and also the analysis. And very similar to the last two. So it has very unique and stable gene expression omnibus accession number, starts with GSE. C stands for the series. And let's look at an example. So this is the overall measles study. Okay, so you can see as you, so on the PowerPoint slide. So it has what kind of expression. So this is Affymetrix, I told you. So the expression binary and what kind of species. And this study was on the heat shock protein 70 and novel axis type 1 interferon dependent antiviral immunity, the measles virus infected brain. Okay, and then the overall design and who contributed to the study and whether it's published. This was published in Virology, Journal of Virology. And then the submission date, and if it's updated, so the date updated, and who submitted, and what institute they belongs to, and then what kind of platform it used, and then what are the samples coming in. So they used 26 samples for this project, and what bioproject it belongs to, and then you can download this information files. And remember, so here it's the place you can download the raw data. Okay, so if you want to start from scratch, you can download from here and reanalyze the whole data, and then probably derive, confirm their conclusion, plus you can identify even more things. Okay, so that is about series. And next is the data set. Okay, let's see, data sets. Okay, so remember, until now, the most of that information is coming from the user. The data sets, those ones are like a specific set of data sets curated by the NCBI staff. The dataset browser looks like this. As you can see here, I don't know whether it's very visible. It says curated. So that means they manually looked at the data and they analyzed it, and they made things available for us. Okay, so this is like very easy to analyze style platform, because they did all the steps for us. And as I mentioned earlier, it's reassembled by the Gene Expression Omnibus staff into gene set records. They have a unique and stable ID starting with GSD. And so it basically gives us a biologically and statistically comparable gene expression omnibus samples. Okay, so and it provide the basis for geo suite of data display and analysis tools. And the whatever the samples within a data set refers to the same platform. Otherwise, it's not going to work now. And also they have already done the background processing as well as the normalization across the whole data set. And there are data set subsets. So there you have the experimental factors that information is given. And so this interface you can basically use without downloading. You can use this interface to see the data, and you can search them inside. And it basically forms the basis for the Gene Expression Omnibus advanced data display and other analysis tool so you can use. Okay, so and one thing you need to remember is not all the series you come across are included in the data sets. Okay, so that's what data sets, and let's quickly look at the data set interface. You can see it's really nice, very well organized, and you can see, so there are data analysis tool, and it will show you what sample subsets include under data set, and also the expression profiles. Okay, so if you want, I can click on this. And remember, it should be in the same platform. Otherwise, it's not going to work. So see, so that's what we are going to look next. So the expression profiles, since they are curated, so you can see the expression of, let's say, your favorite gene across each and every sample. Okay. So here, remember I told you, if you can remember the title. So here they are looking at transgenic mice. That is basically as a modified HSP17, Hitchcock protein 17. And you can see the expression level changes between the transgenic and wild type mice under after specific time frame of infection. Okay, and then the values are given here as well. Okay, so but we will look at it in detail, but this is the overall dataset browser interface. Okay, and let's look at the profiles. So let me change the screen. Okay, so as I showed you earlier, it's the same profile. You can see. So this is the probe ID that corresponds to one specific gene most of the time, and then the across samples. Okay, so they're derived from the data sets, and So it contains the expression measurements of individual genes across all the samples. So here, there are around more than 20 samples under both conditions. You can see the expression levels are given here. So that way, you can get an idea whether your favorite gene is downregulated in one sample and upregulated in different samples. So that information you can obtain. Okay. And you can easily search this profile using the GEO profiles interface. Okay, and then we can look at that interface. Okay, so the same interface I showed you earlier. Okay, so, and these are the values. And you can check individual samples. Okay, so okay, so you can see if you look at this gene, 5 days post-infection, 5 days post-infection, let's say 10 days post-infection, the expression levels, they were more or less same. So therefore, this gene was not very affected. But let me see whether one with the contrasting expression level. Probably, okay, maybe this. Okay, so here, 10 days for measles virus. So here, uninfected. There's no difference, but post infection. Okay, so here you can see, but no, it's you need to, I mean, you can play around and find one with pretty cool changes in the expression. I think you can sort it. Anyway, so you can play around and see. So now it's around 9:40. So we can take another break before we move into completely different section. So you can, I'm going to give you a little bit more break so you can have a cup of tea and come back. So it's 9:40. We will meet at 10, and then we will continue the other half of the lecture. Okay. So meanwhile, I'll stay for a couple of minutes. So if you have any questions, you can ask. If not, you can go for a tea and come back. Okay. Sorry about that. Okay. So before the break, we were discussing about the structure and so basically the interface structure and then the different layers of Gene Expression Omnibus. So moving forward, we are going to shift the topic a little bit. So let me start with the small quiz. Okay, so let me dates and bets. Okay, so check whether you can. I mean, not check, just so please go ahead and answer the quiz, and then I can get an idea about the depth I need to cover. Okay, so I'll wait 15 more seconds and then I share the poll. So it's okay to give answer the the best way as possible. So there's no negative points of extra credit for this, but probably especially during the practicum, we will have graded activities while we are conducting the practicum part. Okay, so 10 more seconds. Okay, so I'm going to end the poll and I'll share the results. Okay, so for the first question, 78% of you got it correct. It's okay to be wrong. And only 59% for the second one. And third question, 78%. Fourth question, 67%. And the last question, 44%. Okay, so yeah. So you guys have a mixed understanding about the file formats. So that's okay. Because I was wondering whether that section was covered by other lecturers. So apparently it's not. So let me stop sharing the results and close this and move forward. So when it comes to the next generation sequencing, we use FASTQ format because, as you are aware right now, the FASTA or FASTA, different people pronounce it differently. The FASTA format, it contains only the sequence information and then the identifier. So that is not enough, because when it comes to the gene expression or genome assembly, the quality also matters, because if you can remember from your Sanger sequencing, I think Dr. Kanishka covered that, the Sanger sequencing part, remember the chromatogram you're getting, all those peaks. So sometimes there are especially at the beginning and end, sometimes if you have a very fuzzy sequence, so there you will see multiple peaks. So therefore you're not 100% sure, because the program is going to call, okay, so this is A, but there can be the minor traces of T or C or G. So then you have no idea about the overall quality. So, but the beauty of the FASTQ, the Q stands for the quality. So it will give you a quality value of your read. Okay, so this is very important, because if you have very poor quality sequences, so then your results will be, it will mislead you because it's not good quality. And if you look at a FASTQ file, as the very last question stated, so there are 4 lines. We will look at each line, and then you will have a good idea about the structure of FASTQ files, because you will be handling FASTQ files in your practicum. Okay, so when it comes to a FASTQ file, you'll see the first line is for the label, where you add information regarding, let's say, your sample, or your what kind of a lane it is sequenced on, and then the sequence information. Usually we get FASTQ files out of next generation sequences, and these sequences will automatically put that information to the label line, so that way we can trace most of the information. And then the second line, it contains the sequence very similar to the FASTA file. And then the third one, so you have a separator. And then the very last one, you have the quality scores. And you will see what is this? AAA at the rate, and then colon, semicolon, question mark. So it looks like Greek, right, for all of us. But actually those are ASCII characters. I will show you the ASCII table. So it will give you an idea, it's very easy to convert those ASCII characters to quality values. So that way the program can easily store that information. So if we look at each line in detail, The very first one, as I mentioned earlier, it is the sequence identifier, and it contains the essential information regarding the sequencing run, and then what kind of a cluster. Okay, and it's determined by the BCL or the FASTQ conversion software used by that specific sequencer. Because remember, the next generation sequencing, you look at like those the if it's fluorometry based method, like in a different fluoropause, and then so you get the corresponding conversion to the sequence, and then the second line. So those are the base call. It can be like the 4 bases or like another IUPAC characters, but specifically here we use ACTG or N. And for like any nucleotide, that means we are not certain that specific portion, the sequence is like A, C, T, O, G. And then the third line is a separator. We put plus sign to separate it. And then the fourth line is the most important part of the FASTQ file. It contains the base quality scores, and they are given in the Fred values. It's Fred plus 32 encoded using ASCII characters, and it will show us the numerical quality scores for our sequences. Okay, so that's the FASTQ file structure. And when you sequence your sample, let's say like if you do Illumina sequencing, you get a file with like millions of these lines because they're like short reads. So and then you can basically like you're not going to look at like in each and every. So you basically use it and then you take it into your alignment software, and there it will be done by the software to like we can trim the bad quality ones before we insert. We will go through all these things in the next week. And then, so basically you can, like everything happens behind the computer screen. So therefore, like we don't need to worry much, but we need to select the correct parameters to do our analysis. Okay, so, and I'm pretty sure now you are curious about the ASCII characters. So this is like an ASCII table used for the Illumina, Torrent, PacBio, even for Sanger. You can see. So each, the quality value, so you have ASCII character. If you look at this column, so there's like a plus, then the colon, semicolon, and remember you saw A and you saw the @ sign. So those are the ASCII characters for the fourth line that we use. Now you are probably wondering how you calculate the Q score. And so there's an equation for that. And Q is equal into -10 log the E of the log 10. Okay, the E is basically the estimated probability of base call being wrong. Okay, so we need the correct one, right? So that's the probability of that specific base to be wrong. And if you have a higher Q scores, it indicates there's a minimal freedom for an error. Okay, so if your sequence run has lower Q scores, that means that results a significant of your portion, the reads are unusable for your next the downstream applications. And because why? It is going to increase the false positive calling of variants, and then you make wrong conclusion. Let's say you take a cancer patient, like a patient, and you take the cancer cells and then the healthy cells, and then you make okay, so this variation happened and this basically increased the oncogenic activity. But later on you realize when you did the analysis, like the sequencing and then the subsequent analysis, your reads were like very low quality. So now, without knowing, because of, it's just actually a mistake, but maybe it's honest mistake. So you didn't pay attention to quality scores. So as a result, you called a wrong variations in, like let's say in an oncogenic gene. And so that will mislead. So therefore, probably eventually you have to retract your paper because it's not correct. So therefore, quality matters a lot. Okay, so in general, a quality score of 20 uh, translate into an error rate of 1 in 100. So that means every 100 base pair sequencing read may contain one error. Okay, so that means one for every 100. That means the accuracy is 99%. And if you think about the Q30, if the value is quality score reaches Q30, that means you can do the calculation. So that is if 99% for Q20, Q30 is 99.9%. So that means virtually all of your reads will be perfect with no errors or ambiguities. And so therefore we consider Q30 as the benchmark quality of next generation sequencing. Okay, so therefore usually when we filter our data, we put okay, so we are going for Q30. So that means your data, although your base calling is 99.9% accurate. Okay, so to give you a very simple way of showing it, it's Q10 means 1 in 10. So that means if you take 100 bases, so there's a chance that you're calling 10 bases erroneously. Okay, but when we jump into the Q20, so it will go to 1 in 100. So that means 99% accuracy. And Q30 means 99.9% accuracy. And if you want to go even more stringent, you can go for Q40. Okay. So this is how you determine the quality of your samples. Remember, because if you're planning to submit your data into Gene Expression Omnibus and then this small strict archive, it's always good to have a better, higher quality sequencing data set. Because sometimes we like it or not, so we have to dump our data because they're poor quality. Can't help. Because usually, when we do whole genome assembly, one sample is going to cost around 50,000 to 60,000 if you send it to Macrogen. But if you send it through the local agent, it is going to cost 180,000. Okay, so because when we send it through the gene tech, it will cost us 180, around 180. But if we send it directly after getting permission, it will cost only 60. So therefore, that kind of thing is also there. So therefore, we always try to get the permission from the relevant authorities to send sample directly. So that way we can do 3 samples instead of 1 sample because 60 multiplied by 380, right? Yeah. So therefore, we can do 3 instead of doing 1 through the gene tech. Okay. So unfortunately, I mean, it's very unfortunate for local researchers. We always pay huge premium for the local agents, and sometimes it's very unfair because the same thing we can hand carry that, let's say, equipment for 1/3 or sometimes 1/6 of a cost compared to buying it from a local agent. So we are not developed to have a infrastructure like Singapore, even Thailand or Bangladesh. So that is kind of the sad reality. Probably you guys will go and join bodies like the policy making, NMRA, and other places. So make sure to try to understand this issue and come up with a solution where all of us can do quality research with the limited budget we have. Okay, so moving forward. So now you have a good quality data set. So the next thing is how we are going to submit it to the Gene Expression Omnibus. Okay, so sometimes it can be a little frustrating because there are so many. Although remember, at the very beginning, I mentioned that NCBI wanted to have a very user-friendly, very easy, very simple way of entering data, but sometimes still, even with all this simplification, sometimes can be like a little frustrating. Okay, so the data. So now, like the thing is, like you need to flow your data into the system. Okay, so you start with raw data, and then you process it, and then you have the processed data, and then you have you have to put the metadata, and then you submit it to the Gene Expression Omnibus. Okay, so this is very simplified way of the things we do when we submit our data, and it's very important to have a good quality raw data and near perfect pipeline to analyze data, but it's also very important to have a good metadata. Remember, we talked about the Miami and MinSig guidelines. So you have to have good metadata, so that will make our life very easy. Not only our life, the NCBI staff, and also the researchers who are going to reproduce our data. Okay. Okay, so let me quickly check whether you're awake. Okay. It's a very simple question for you all. From the previous, the first half of the lecture. So I'll give you 1 minute to answer. The answers are coming, so that's why I'm waiting a little bit. Okay, 29, that's not a bad 54%. Okay, so I'm going to end the poll, and I will show you the results. Okay, it's only 63% of you remembered what we discussed during the first 2 hours. Okay, so but it's okay. So you can always go and check. The answer is GSC. And 63% got the answer. Anyway, so let me stop sharing. And we're moving forward. So we were discussing about submitting data. Okay, so when you start the submission process, first you need to prepare your raw data. So this is general overview. I'll give you more details in a bit. Okay, so for the next generation sequencing, you use FASTQ files, and the arrays, we have the cell files, or GPL file, not cell files. So there's another one I can't remember. And then so you have here you're using original unprocessed file, and also you need a processed data, the expression matrices, and whatever the normalized values of your genes, and then the overall results. And as I mentioned earlier, metadata is most important. So there you should have your sample descriptions ready, experimental protocols ready, and then you check whether you're fulfilling or satisfying Miami or the MinSig data requirement. And then you can upload it via the web interface. And then the NCBI will basically give you a temporary number or temporary accession number while they look at your data and see whether you are basically in compliance with this minimum data requirement, and are there any missing information related to other aspects as well. And after maybe sometimes last time when we did submit our results, it took 2 to 3 weeks for them to give us the final say, okay, so your data set is fine, and now you can get a proper accession number. And remember, so sometimes when we before we submit our manuscript of our paper to a journal for the reviewing, we need our accession number. Okay, so but we don't want our data to be public. because even before we publish, because some have they have more infrastructure, they can reanalyze our data even before we publish, and then they can publish. So therefore, we are always given the choice that we can say in CBI, okay, so this is unpublished, so therefore hold our data for 6 months or 1 year. Sometimes even you can request up to 2 years. So then they will not release, but they will give you an accession number and then the confirmation that you already submitted your data to the NCBI. So that way your journal will going to be okay with your data set. Okay. And the thing is, you can hold your data until you publish. So let's say you put 2-year embargo, but if you publish paper, your paper citing your, the accession number after 6 months, so then the NCBI can trace it and they will release your data to the public. Because, so once published, you can't hold your data. Okay, so that is the condition uh, they put on us, and it's perfectly fine because once you publish, people should be able to access your data. Okay. And always remember the raw data is sacred and the metadata is very important, and you have to have proper documentation. It's our submission mantra for the high throughput genomic data. Okay. So let's see whether there are any questions. There are no questions. We are moving forward. And so there are different data options we can take when we submit our data. And you already know what those two data options are. One is the microarray or next generation sequencing. And also this is, yeah, you can use this same for the non-coding RNA as well, because there are more than 30, I heard around 300 different RNA types. Those are not coding, non-coding, and those ones can also be submitted as microarray or next generation sequencing, because they will not translate it into protein, but rather they stop at the RNA level. And also, if you're really interested in doing promote analysis, I'm that's part of my research. I study the transcription factor and then the cis element, the promoter interaction, and how it changed the gene expression level. So one of the techniques we use is what we call as the chip assay or the chromatin immunoprecipitation. And those data is also high throughput data, and they can be submitted as both microarray and next NGS sequencing the platform. And also there's some people study the epigenetic changes other than what I mentioned earlier. So the methylation assays. So those ones also can be submitted as a microarray or next generation. And also some people, especially under clinical settings, people use high throughput reverse transcription, PCR, to identify the different genome variants, or what we call as RACGH. Those data can also be submitted. And also you have the option to select SNP arrays. And because especially if you're doing GWAS, I don't know, you heard about GWAS, genome-wide association analysis. You do, let's say Sri Lanka, chronic kidney disease used to be one of the major issues, and still it is. And then so in a couple of groups, what they did is they wanted to see what is the genetic basis of this, the genetic composition, how it affects these patients. And they identify a couple of risk loci. So that is done through the SNP arrays. So that can be, those data can be also uploaded into the GEO as a data option. And also SAGE, that's a serial analysis of gene expression, and even protein arrays. So those are the data options they are given. So you, based on your experiment, you need to pick the correct data option for your submission. Okay, so now you know the options. So let's look at different requirements. Okay, so you have to have raw data, process data, and metadata. So I'm re-emphasizing this. I mentioned this one twice already. So this is very important, especially when it comes to the submission, raw, process, and most importantly, metadata. And putting raw data, it facilitates the unambiguous interpretation of the data you generated. And also it will help others to verify your data and see whether your conclusions are correct. And when it comes to the microarray data, so you can provide microarray data within the sample record data table or as an external supplement file. I think you saw the CEL file type. So you can provide one of either sample record data table or the cell file. And for the NGS sequencing that we are doing right nowadays, so here the geo it brokers a complete set of your data files. Remember I mentioned about this SRA, sequence read archive. So they basically deposit your raw data into sequence read archive on behalf of you. Okay, so therefore you don't need to upload it to SRA and then upload it to geo. No, geo just take everything and then they put the raw data into the sequence read archive on behalf of you. Okay, and you need to satisfy the compliance to Miami and MinSeq. So those are the requirements when it comes to submitting data to Gene Expression Omnibus. Okay, so remember, one of the major goals of this Gene Expression Omnibus was to provide easy access to this database and keep it simple, what we call as the KISS, keep it simple and stupid. Okay, so the KISS principle applies to this one as well. And they provide templates for us to use. Okay, so and make sure to provide a valid email address because they communicate with you through email. So therefore, it's essential to have a valid working email address when you submit. And also, if you're working for an institute, so you can provide your institutional email address. Sometimes people prefer to put their personal address because sometimes you move from one university to another. So then you sometimes lose your access to your email. Okay. And so they have templates for microarray. So there are both one color microarray and two color microarrays. So depending on that, you have different templates you can use. And for high throughput sequencing, so there is a metadata spreadsheet you need to provide. So that will basically the program or their database will automatically identify, okay, so this is the field and this is the description, and then they will automatically put it into the correct place. And when it comes to the human research, okay, so we have to satisfy the ethical review compliances as well, because we can't just expose human data. So for that, they have there's a specific database what we call as NCBI DB gap database. So that is for human subjects, where you can incorporate confidential information regarding the whatever the cohort you're using, but it's restricted for other people. So therefore, there's the limitation. There are limitations when others access this data, because we need to protect the personal identities when it comes to the human research. Okay, and when we look at the benefits, okay, of submitting our data to Gene Expression Omnibus, as you're already aware, so you'll get a very unique and stable accession number. And so this number you can use for your manuscripts. Okay. And as I mentioned, the data can be held private you can have an embargo period. So therefore, others won't be able to access until you publish your results. And the journal or the they call article review process, where so where you submit your manuscript, and then independent review, sometimes blind review, sometimes open review. The people will review your paper before you publish. Okay, so and they should get access to the record, and the NCBI is provide the reviewers to anonymous access to those private records, so that way they can verify this is not a fake study, because nowadays that is one of the issues. So we have so many predatory journals, and then there are so many shady, the not shady scientists, but it's not, I mean, scientists can't be shady, so therefore there are no shady scientists, but there are some fake scientists, and they're trying to basically make a lot of fabricated studies. Those things can be verified because of these systems. And also, you have the freedom to update your records. Sometimes you add additional information. If you can remember, when you looked at our samples, you see upload date 2004 June something, and then updated on 2019 November something, likewise. So therefore you can continue. You need to send them an email and say, okay, so add this additional information. So then they will do that for you so that way you can keep your data set up to date. Okay, so that is another advantage or the benefit of using Gene Expression Omnibus. Okay, and there are some limitations. Okay, so Gene Expression Omnibus won't accept human data. As I mentioned earlier, you need to submit it to a dbGaP and control access small reads archive. And it do not accept transcript assemblies. Okay, so those ones need to be submitted directly to the SRA. Or if you use shotgun sequencing, you need to add it to the transcriptome shotgun assembly. And whole genome sequencing, so you can directly submit it to the small rate archive and WGS. And if you're doing metagenomic sequencing, again, you need to directly submit to the SRA. And if you're doing resequencing, variation, copy number analysis, again, it's SRA. And same for the service sequencing and whole exome sequencing. Those data will go into different databases than Gene Expression Omnibus. It makes sense because when the Gene Expression Omnibus was founded, so those days we only had microarray data. So the gene expression data and some proteomic data. So therefore, those days we didn't have that kind of specific requirement, but the databases and our research process both are evolving. So accordingly, we have now multiple databases and very unique databases for unique set of data sets. Okay, so that's what happened. So now you know the benefits and limitations. So let me quickly challenge you with a question. Okay. Oh, perfect. 86% got it correct. So in general, it requires both raw and processed data, but there are some very rare instances. They you only need to submit raw data, but most of the time, so you're supposed to submit both raw and processed data. Okay, so therefore, in generally, the both raw and processed data are required. Okay, so let me stop sharing the results and close it. And moving forward, so when it comes to depositing your data and accessing data, because sometimes you need to upload more than a terabyte worth data. So therefore, like, you have to have, depending on your size of your files, you need to come up with a different strategy. There are different means to do it, especially for microarray. We just do the direct upload because those files are like, tiny compared to the other files. And for the large data files, you can use a FTP client like FileZilla. Okay. And so you can just like, let it like, work on the background while you're doing other activities. And the good thing with this is you can even like, pause your data transfer and then resume afterwards. Okay. So it won't break the file if you like, pause the transfer. So because remember, like, so here, like, we don't have so-called real unlimited data. And what is going to happen is like, you run out of your data quota when you try to upload your data. Or like, if you think about our university connections, they are of course good. Like, we have like 100 Mbps line, but the thing is like, every 5-6 hours something happens and then it get disconnected, and you don't want to lose like whole progress you like done during like 5 hours of your time. And so therefore, like this kind of a FTP transfer is more appropriate compared to our situation. And also they have their own tool called Aspera Connect that uses like, not FTP, but UDP, or use a datagram protocol. And that can also be used to upload your data. Okay, so FileZilla is free. I think they have a pro version as well, but you don't need that. You can use these three methods. Okay, so when it comes to downloading data from the Gene Expression Omnibus or SRA, you can use FTP to download it, and you can use the accession display biology. That means like direct download, and also you can use URL download, and you can even do the programmatic access. There's a set of tools called interest program utilities or eutils, and that you can use. And also you can do the dataset query and download it. So you can like, when you go there, you can see the different approaches you can take in order to upload as well as to retrieve your data from Gene Expression Omnibus. Okay. To basically like, simplify all those methods, you can use the web interface. You can use command line tools. Like, I'm pretty sure some of you are very familiar with the Galaxy interface. So there you go and like download the data sets using FASTQ dump. It's very easy. So then it will do the job for you. And also you can like use bash scripts and like various tools. And also you can use program interfaces like R. There are different bioconductor packages coming under the R to do your job. And also you can use other means like Python libraries, and then there are other automated analysis. Okay, so the trick is you need to pick the correct approach based on your data style and also the computational skills. I don't know how many of you are familiar with the command line tools, because most bioinformaticians tend to use Linux or Mac OS for their work because it's easy compared to the Windows interface. And so there are some programs like my preferred one. I mean, I use both Mac and the Mac, Linux, and Windows interface. So usually I use the tool called MobXtream to like access the supercomputer. So that provides a Linux-like environment to the Windows interface. And also, if you're very familiar with, you can use Windows PowerShell as well. So now Windows also moving towards more like Linux side, considering all the advantages you can have. Okay, so you can like try all those approaches. If you're not really good with the script, but now you have all those AI tools, So therefore, like you can basically generate a good script without much hassle. Like before AI period, like we had to struggle and troubleshoot. There are like different websites like BioStars and Seek Answers. They like goes, okay, oh, my program is giving errors, so can you help us? So I mean, we had to like go through many sites in order to get good help. But now with the now we're in the era of AI, so therefore like that it makes your life easy, but doesn't mean you can forget fundamentals, because otherwise you will become an well, sorry to use the term, like an idiot, because the AI will think, but you're not going to think. So that way, you are not going to understand the real the importance behind your data set. So therefore, always, I'm never discourage a use of the AI. I also use it like most of the time, but use it wisely. So make sure AI is not going to make you an idiot, because I'm pretty sure you have seen like in the movies where like an AI will take over the world. So you don't want that, right? So therefore, like use them wisely. Okay, so when it comes to search, so there are various tricks and tips we can use in order to search the data from Gene Expression Omnibus. Okay, so we will have like a separate practical part for this during your practicum. But until then, so let's see what are the possible or what are the available options you can follow in order to search and retrieve the most important studies for you. Okay, so like Google search, when we do Google search, we just do the free text search. You type like let's say smoking cancer, and then you get all the whatever the accessions or data sets with that specific like term smoking or cancer or smoking cancer. And if you want to make it like less non-specific, let's say like it can be like some people write okay, the effects of smoke, and some people write it as smoking. Okay, so then you can use a wildcard. The asterisk is a wildcard, so that means every word start with smok will get as a result when you use this kind of wildcard. And also you can use this boolean expressions, right? Like O and not. So those ones you can use in order to restrict. So smoke or diet means either smoke or diet or both will appear, and means so the first two terms plus this whatever the mammals, the organism, and you can see within square brackets we have organisms, so that way we can restrict only to mammals. Okay, but not to humans. So mammals can take many species, including human. But let's say you don't want human, so you use this not expression. Okay, and also if you want something from a project, so you can use that qualifier. Okay, and then you can sort based on the study type, and even you can define. Okay, so this the data sets with 100 to 500, not more less than 100, not more than 500. And then you can also define what kind of a cell type, like a supplementary file type you need. And let's say it's from you, like let's say it's authored by me. So then it's like Herat author. Now there are many Herat. So therefore, like you will get one. I can even further specify when Herat. So, and also publish before, between, or after that we can decide, and also restrict or filter, and also we can do it based on the platform. Okay, so therefore a lot of flexibilities because if you go to here, and let's try one. Okay, so I want all of you to go to I will share the link. geo data sets. Let's see, everyone. Okay, so go, visit this site and then try this search and let me know, use the chat box to show me how many studies you got as a result. Okay, so you can paste your results on the chat window so that I will know how many results you got and whether there are any differences between 50, how many of you online, let's see, 51. Okay, so let me make it a quick Zoom poll. Let's see. Okay, let me launch it so you can answer. You can use this instead of the chat window. I feel like you guys are shy to use the chat window. So therefore I created a small Zoom poll question. Okay, you can just put the number and submit. Oh, sorry. I don't think you, okay, my bad. You guys didn't tell me no. So this is the search term. Because I'm sharing the desktop rather the program. Very interesting. You'll see your those who are on the chat window, you can see some your numbers are not tallying with the others because look very carefully. If you miss one bracket, one square bracket, so the results will be completely different. So therefore so make sure depending on your requirement, you have to have the correct search term because missing one side of a bracket is more than enough to give you completely different number compared to the other person who's having all all sides of the brackets. Okay, so far we have 300 values, 400 value, within 400 there are 445, 467. So likewise, because all of you are searching against the one database and getting different values. This is really cool. Okay, so I'll give you 15 more seconds and then okay, so we have 285, 377, 445, 191,842, 367, 343, 445, 460. Okay, so likewise, many results. Okay, so let me type the close it. The same COVID. Oh, sorry. So even missing a letter can change everything. COVID-19. Okay. So I can see there are 460. And so you will see you can cross check with your expression and then see why why it was different from the answer I got. And from here you can see yes, there's a mouse, rat, macaca. That's a monkey, right? It's a primate, yeah. So likewise, uh, you get the organisms all other than, you can expand it here if you want. you can see everything other than human, us. Okay, so play around and then see what kind of things you can discover. So you can, let me go back. So you can do same kind of thing for the gene expression omnibus profiles as well. Earlier it was data sets, and also you can look at the profiles. And remember, there are different fields because we already discussed about the structure. And as you can remember, so there are different fields. So based on that, you can define specific search terms. So this is something we are going to do during your practicum as well. Okay, so and I will give you activities, and there will be modal submission, etc., etc. So to do this kind of a search. Okay, so moving forward, now you have data. But remember, I told you, NCP, I thought, okay, so it's not only grab raw data from there and do analysis. So let's provide something to this data and insight with this data. Okay, so then only you can make some kind of meaning out of this data. And then to do that, as I mentioned earlier, you can take the raw data and do the analysis, or you can take the lazy man's path. This is how you do exercise, right? Very effectively and very lazy manner. Not effective, but looks like. Okay, anyway, so to do that, so we have a tool called Geo2R. It's your online analysis sidekick, so that you can use to compare group, to do the statistical analysis, to visualize the data, and eventually export the data or export the results of your analysis. Okay, so basically it's inbuilt tool you can use to do your data analysis. It's very simple. You select data sets and then you define the groups, and then you run analysis, and then you download the results. Okay, so this interface is a very, I can't say it's primitive, but it's very powerful, yet very simple thing you can use. Okay. And to give you some more insight to Geo2R, as I mentioned earlier, it's an interactive web tool, and you can use it to compare more than 2 samples. And also, you can use this to identify genes differentially expressed across experimental conditions, and it will give you a tabular and graphical output that you can use for your publications, for your reports, for your projects, likewise. And it uses, it stands for Geo2R, right? So that means it uses the R environment. And so specifically, the bioconductor packages of the R, because the bioconductor is a curated set of plugin that we can use for genomic analysis. There are very, very, it's open source, and you have large number of tools that can use for any analysis. Okay. And so for microarray data, so it used these 2 bioconductor packages called GeoQuery and Lima. And for the RNA-seq, it's still at the beta stage. They use DSeq2, the very popular differential expression analysis tool developed by Michael Lau. That's his name. And it's very, very nice person. So if you ever use DSeq2, if you have any questions, when you pose a question, he himself answers the questions. Okay, so that's how nice he is. Okay, so I will show you a video showing how to use Geo2R, like in a very, very simple tutorial. But before that, so you need to you should be able to make sense of your data. Okay, so before you do any analysis. Okay, so the process include like data import, normalization, integration, and analysis. Everything of this is happening behind the geo interface or like a geo2r interface. Okay, there are some challenges when you integrate data from coming from different platforms and different protocols, because sometimes they won't tally. So then you get a result. That's the thing with computer program. When you input something, you will get some kind of an output. The question is, as a scientist, whether it's scientifically sound and statistically correct. So that you need to check. And there can be batch effects and biases. Those need to be excluded. And there's a chance that there are missing or inconsistent metadata. So therefore, you can't use it. So like, the meta-analysis I was mentioned earlier, we are doing with one of your seniors. So they are also like, we have like hundreds of data sets, but unfortunately, the metadata is not very comprehensive. Therefore, we can't use it. We have to exclude it. And also there's a issue that the data, they can be in different format. So then they are not cross-compatible. In that case, you have to address this issue as well. Okay, so the next, I'm going to show you a tutorial. This is the link. I will share the link, but I have the video as well. So let me play the video for you. Please check.
This video updates a 2012 video called Geo to R, Analyze Geodata. Both videos demonstrate how to use Geo to R to analyze gene expression data present in Geo. Now more than 99,000 studies are available for analysis with Geo to R. Plus, we've added 2 new features, the ability to analyze RNA-seq data and interactive visualization tools to help you explore the data. In this demonstration, I'm going to analyze the GeoSeries GSE 41586, which investigates gene expression in a human colon cancer cell line. There the cells are treated with 3 concentrations of the chemotherapy drug 5-aza-deoxycytidine. I'll begin the analysis by scrolling down and clicking the button Analyze with Geo to R. Before I run Geo to R, note the full instructions link near the top of the page. This documentation includes edit options and features that are not part of this video, and provides references for the software that forms the basis of the tool. And to be sure to read the section detailing Geo to R's limitations and caveats. Going back to Geo to R, we see a list of the samples in the study, along with metadata extracted from the original records. The samples are grouped by the 3 treatment levels, 0, 5 micromolar, and 10 micromolar 5-aza. The first thing we need to do is to define the groups of samples that we want to compare. Click Define Groups and type a group name into the box. You can name the groups anything you want. I'll type Control, hit Enter, then Low Dose, Enter, and High Dose, Enter. Next, we need to assign the samples to each group. I'll click on a row and drag the cursor over the relevant samples. Then click the group name that they belong to, and repeat for each group. Note that Geo2R supports up to 10 groups. I can now run the analysis. Down the page, I'll click on the Analyze button. This performs a comparison with default settings, and after a brief wait, I see several visualization tools, and below those, a table of the top 250 differentially expressed genes. FC1 is more highly expressed in the low-dose group compared to the other 2 groups. You could also use the Profile Graph tab to explore a specific gene. Here you enter either the gene symbol or gene ID number, which you can get from the table, or by downloading an annotation file with this link, Download Gene Annotation. For example, if I want to see the graph for nitric oxide synthase 3, I enter either NOS3 or the ID 4846. Clicking on Set displays the graph. I'll now go back to the table of the top differentially expressed genes. I can edit this table by using the Select Columns feature. This opens a dialog box that lists all the available statistics and annotation columns. To save the full set of results, use the Download Full Table link to export the results as a tab-delimited file. Now let's take a look at the new visualization tools. We see 7 different plots for visualizing the data. Let's look first at the UMAP plot. Click on the plot to open a larger pop-up for better viewing. In general, samples clustered together are more closely related than those found farther apart. See the full instructions that we saw earlier for more explanation. For this series, we see that the samples cluster within each group, but the groups are not closely related to each other. If I want to look at the relationship between samples before running an analysis, click Analyze before assigning samples to groups, and the UMAP plot and box plot will be displayed. The plots outlined in green have interactive features. I'll click on the first plot on the left, the volcano plot, then on the Explore and Download button. Here red dots represent genes with increased expression and blue with decreased expression. Data below the threshold is colored gray. Scrolling over points on the graph reveals gene information, the log2 fold change value, and p-value. Radio buttons at the bottom of the window allow you to toggle between comparisons, called contrasts here, and you can download a tab-separated file containing data for the selected contrast. You may want to change test parameters and run the analysis again. Use the Options tab to make changes to the p-value adjustment method, or significance level cutoff, or log2 fold change threshold. I'll lower the significance level cutoff to 0.001 and the log2 fold change threshold to 2. Then I'll click Reanalyze. Now some of the plots are revised, including the volcano plot. Finally, the RScript tab displays the script used to perform your last analysis. You may want to save this as a reference. That's all for this demonstration. If you have any questions or feedback, please email the GEO team, geo@ncbi.nlm.nih.gov, or use the Email GEO link at the top of the page.
Okay, so I lost power for a bit. Hopefully you guys didn't miss some parts of the video. But anyway, I mean the link was also there. So next, this is like a pretty cool thing to work with, but we need to think about the limitations as well. And so there's a big chance that whatever the results you're getting are not going to be equal with the publication results. So that is possible because so here the data flow, remember, let's say microarray data, it's a GeoQuery versus Lemma platform, but we can use different other tools as well. So they follow different algorithms. So based on that, the results can be slightly different. So that is a possibility, but doesn't mean the earlier one is wrong or your one is wrong. As long as you use the correct parameters, there can be variations because the assembly parameters can be different. Okay, and also there's a chance that some of the samples are missing when it comes to the RNA-seq, and also sample value mismatches can happen. In addition to that, there can be data type restrictions like the microarray, the value column of the sample tables. So they are not available. So therefore, you are not, you don't have access to do check those values. Okay, so that is another issue. And sometimes when the value is not at all given, so then you can't use it for the analysis. You can always go to this link and then read about other limitations of GeoToR. But it's a very good place to start your journey of analyzing data from Gene Expression Omnibus. Okay. So you guys have any questions regarding the things we discussed so far? And you can, if you want, you can unmute yourself or you can use the chat box. So you can ask. And that's all I'm going to cover for today. And next week, we are going to discuss about the genome assembly, like the principles of genome assembly. And we will probably do like some small activities before we get ready for our big practical or the practical. Okay. So I will stay here for like another 5 minutes or so if you have any questions. And if you don't have questions, so you guys have like great rest of the weekend. Thank you.
Excuse me, sir.
Yeah.
Sir, I have a question, not regarding this specific lecture, but for this course, for our case study, we are having a computational part where we are doing cross-species comparison using NCBI's comparative genome viewer. Like we got the result, we can generate the results for the species we want, but we don't know like how to meaningfully interpret the results, as in a way we can put it into our like final report, as in like the genome viewer gives you the visual result, but not like the ratio of the similarity, the synteny. Like we don't know what to do regarding.
Yeah, so I mean, so basically, like you're doing this internet analysis, right? Like specifically focusing on one specific region or the whole genome.
The whole genome. We want like a percentage of how similar they are, so we can have an idea like what genome is more compatible with human genome than the other genomes. So in order to do that, we need to have like some data to go off like the percentage.
Yeah, so I mean, there are like a couple of tools. So like personally, like I don't use that interface, but rather I download the genome and do the analysis. So like the MC Scan X is like I think the most popular one. So that will give you like all these the inversions and like duplications, like all this matrices you want. So that is one approach you can try in addition to whatever the approach you're taking. So because it will give you like an overall view. And also, there's a one called Koji, C-O-G. Maybe you already used that one.
Yes, I did, sir. It gives the dot plot view, right?
Yeah. So and Koji, it will give you more information as well, like the what do you call again, like the duplication related things and like the inversions and because since you're like comparing the whole genome, because we are mostly focusing on specific region, so then we are like mostly focusing on like what are the indels, like the duplication likewise. So MC Scan X is a good one that you can see in order to get an output. I'm thinking what other tools you can use. So like whatever your assignment, so what is what are the questions they're asking?
I know it's our own proposal.
Your
You
own
are
proposal.
not given assignment or anything like that.
I see. I got it. Yeah. So so when is the deadline? Like when are you planning to submit this proposal?
Since the exams are coming on August, we are planning to finish the study by the beginning of August.
So I mean, I can like give you some guidance. So if you can either send me an email or like I can what else I can do. Yeah, so email I can give you like basically like how to summarize your data, like some guidance, summarize your data, because out of my head I can't remember like the exact things I use because I didn't do a synteny analysis for like 2 years or so. But definitely like I can give you some assistance. So like just shoot me an email and I will send you more information so that you can use.
Okay, so some guidance would be great. Then I will email you.
Okay, yeah, cool.
Okay,
And
thank
I
you
think
very
you have
much.
my email, right? It's venura@agri.pdn.ac. I will type it here in the chat box.
Okay, sir. Thank you.